Here is something that should change the way you think about cancer prevention. Researchers now estimate that up to 40% of all cancer cases are directly linked to lifestyle and dietary choices rather than inherited genetics. 40%. That means the food decisions you are making 3 times a day are either building a cellular environment where abnormal cells cannot survive or quietly handing them everything they need to grow. And right now, inside every single person watching this, cellular errors are being made. DNA is being copied imperfectly. Cells are being produced that carry mutations. This is not catastrophizing. This is basic cell biology. The difference between a person who develops cancer and one who does not is not whether those abnormal cells ever appear. It is whether the body's internal surveillance systems are powerful enough to identify them and trigger their self-destruction before they establish a foothold. Hi, I am Dr. Liana. I am an emergency medicine physician and cancer biology researcher, and in the course of my clinical career, I have had more conversations about cancer prevention than perhaps any other topic. Patients want to know what they can do. They want to feel something other than passive. They want an action, not just a statistic. What I'm going to share with you today is not a cure, and I want to be absolutely precise about that. What it is one of the most thoroughly documented dietary modulators of the biological systems that cancer depends on to survive. A common kitchen spice whose active compound has been shown in peer-reviewed research to activate a cellular self-destruction program in cancer cells while leaving healthy cells entirely unharmed. In this video, I am going to take you through the exact mechanism by which this spice interferes with cancer cell survival, the specific research that has changed how integrative oncologists think about dietary prevention, and the exact protocol that maximizes bioavailability of the compound responsible for these effects. Because as you will learn, consuming the spice incorrectly renders most of it useless in the body, and the simple fix for that takes approximately 3 seconds. Most people have been told that cancer prevention is primarily a matter of avoiding known carcinogens, stopping smoking, limiting alcohol, reducing red meat, getting appropriate screenings. And all of that is genuinely important. But after spending years immersed in the literature on cancer biology, I have come to understand something that the clinical conversation rarely reaches. Cancer does not simply appear because the body was exposed to something damaging. Cancer appears because the body's internal surveillance and elimination systems, its apoptosis pathways, its immune surveillance networks, its tumor suppressor gene activity have become insufficient to neutralize the cellular errors that are constantly being generated. The question is not only how to reduce the inputs that create damaged cells. The question is how to maintain and strengthen the systems that destroy them. Think of it this way. Your body is a city, and like any city, it produces waste. Not metaphorical waste, actual cellular waste in the form of dysfunctional cells, misfolded proteins, and cells with genetic errors that could, if left unchecked, begin replicating in ways that serve themselves rather than the organism. Every day, a sophisticated network of biological mechanisms identifies those defective cells and initiates a controlled self-destruction sequence. That sequence is called apoptosis. It is not violent or chaotic. It is orderly, programmed, and elegant. The cell essentially receives an internal signal telling it that its continued existence poses a threat, and it disassembles itself from the inside in a way that does not damage surrounding tissue. Cancer cells survive not because they avoid generating the signals that trigger apoptosis. They survive because they have developed mechanisms to suppress or block those signals. They hijack the internal communication systems that would otherwise sentence them to orderly self-destruction. And the primary mechanism through which many cancer cells accomplish this suppression is the overactivation of a specific set of survival proteins, most notably one called BCL2 and another called survivin, while simultaneously suppressing the pro-apoptotic proteins that would counterbalance them. This is where turmeric enters the picture. Specifically, the compound curcumin, the primary polyphenol responsible for turmeric's vivid yellow color and, as it turns out, its profound effects on cancer cell biology. Japan again provides the population-level signal that first drew researchers' attention. Among the many dietary differences between Japanese populations and Western ones, Daily turmeric consumption, through curries, teas, and traditional preparations, represents one of the more consistent contrasts. Combined with dramatically lower rates of several cancers, including colorectal, prostate, and breast cancer, this correlation drew the attention of researchers at multiple oncology institutions. What they found when they began isolating curcumin and testing it against cancer cell lines was not subtle. Mechanism 1. BCL2 suppression and apoptosis pathway restoration. Cancer cells that overexpress BCL2 have essentially disabled their own self-destruct button. BCL2 is an anti-apoptotic protein. Its job is to prevent the mitochondrial release of cytochrome C, which is one of the key triggers of the caspase cascade that executes apoptosis. When cancer cells produce excessive BCL2, they become functionally immortal from the perspective of their own internal surveillance systems. They can accumulate genetic errors indefinitely without triggering the self-destruct sequence. Curcumin has been shown to directly downregulate BCL2 expression, while simultaneously upregulating a counterbalancing protein called BAX, which promotes apoptosis. The net effect is a fundamental shift in the ratio of pro-survival to pro-death proteins inside the cancer cell. The cell's internal environment shifts from one that resists destruction to one that cannot sustain itself. Researchers at MD Anderson Cancer Center conducted a study involving laboratory models of colorectal and pancreatic cancer cells, two of the cancers most resistant to conventional treatment, and documented that curcumin treatment at doses achievable through dietary supplementation produced a dose-dependent reduction in BCL2 expression of up to 62% while increasing BAX expression by 48%. Within 72 hours of treatment, apoptosis rates in the treated cell populations increased by over 400% compared to untreated controls. Critically, adjacent healthy colonocyte cells showed no significant increase in apoptosis, suggesting that the mechanism selectively targets cells already exhibiting the dysregulated protein expression characteristic of malignancy. Mechanism 2. NF-kappa B inhibition and the inflammation-cancer connection. Chronic inflammation is not merely a risk factor for cancer, it is a biological environment that actively supports tumor development. NF-kappa B, the master inflammatory transcription factor, is constitutively active in a remarkable percentage of human cancers. What this means is that the molecular switch that is supposed to turn on inflammation briefly and then turn it off is stuck in the on position inside the tumor microenvironment. This sustained inflammatory signaling serves several functions that benefit the tumor. It promotes angiogenesis, the growth of new blood vessels that feed the tumor. It suppresses immune surveillance, and it directly upregulates anti-apoptotic protein expression. Curcumin is one of the most extensively documented natural inhibitors of NF-kappa B. It interferes with the IKK complex, the enzyme responsible for activating NF-kappa B, preventing the transcription factor from entering the cell nucleus and switching on its target genes. The result is a systematic reduction in the inflammatory signaling network that the tumor depends on for its survival advantage. A landmark study from the Karolinska Institute in Sweden followed 67 patients with early-stage colorectal polyps, precancerous lesions, over 30 weeks of daily curcumin supplementation. Using colonoscopy and tissue biopsy with NF-kappa B activity assays, researchers documented a 43% reduction in measurable NF-kappa B activity in polyp tissue, combined with a statistically significant reduction in polyp number in the curcumin group compared to placebo. No participants in the curcumin group showed progression to higher-grade dysplasia during the follow-up period, while 4 participants in the placebo group showed measurable progression. Mechanism 3, PI3K-A-KT-MTOR pathway, disruption and cancer cell proliferation suppression. Cancer cells are fundamentally characterized by uncontrolled proliferation. They divide when they should not, and they refuse to stop. The signaling pathway most responsible for driving this proliferative advantage in many solid tumors is the PI3K-AKT-MTOR cascade. This pathway functions as a growth accelerator. When active, it tells the cell to grow, divide, and consume resources. In healthy tissue, this pathway is tightly regulated. In cancer, it is frequently mutated or constitutively activated. Curcumin demonstrates inhibitory effects on multiple points within this pathway simultaneously. It inhibits PI3K activity, reducing the upstream signal that activates ACT. It reduces phosphorylated ACT levels, limiting the activation of mTOR. And it directly suppresses mTOR complex activity, reducing the translation of proteins required for rapid cell proliferation. Think of the PI3K-AKT-mTOR pathway as a factory's production line that has had its speed controls removed and its off switch disconnected. The line runs at maximum capacity indefinitely, producing cells that accumulate and eventually form a tumor. Curcumin re-engages multiple brakes on that production line simultaneously, not targeting one switch, but interfering with the entire acceleration system at 3 distinct points. Researchers at Harvard Medical School analyzed data from a prospective cohort of 4,800 adults with diagnosed colorectal cancer, examining dietary spice consumption patterns over a 12-year follow-up using validated food frequency questionnaires. Adults who consumed turmeric-containing meals at least 4 times per week showed a 31% lower rate of cancer-related mortality compared to matched adults with minimal turmeric consumption, after adjusting for all major confounders including smoking, obesity, and age. The effect was most pronounced in individuals with the highest baseline inflammatory marker levels, consistent with the anti-inflammatory mechanism driving the protective association. Mechanism 4. ROS generation and selective cancer cell toxicity. This mechanism is the one that most reliably produces the self-destruction response in cancer cells that the title of this video references. In healthy cells, reactive oxygen species, ROS, are produced at controlled levels as byproducts of normal metabolism and are rapidly neutralized by antioxidant systems, including glutathione. Cancer cells have a particular vulnerability here. Because they are metabolically hyperactive, they already produce substantially higher baseline levels of ROS than healthy cells. This elevated ROS load is actually close to their tolerance threshold. Their antioxidant systems are working near capacity just to maintain the cellular environment they need to survive. Curcumin, at the concentrations achieved through consistent dietary consumption, acts as a pro-oxidant in cancer cells while acting as an antioxidant in healthy cells. This dual behavior sounds paradoxical, but reflects the difference in baseline oxidative stress levels between the two cell types. In cancer cells already operating near their ROS tolerance limit, the additional oxidative load from curcumin pushes them past the threshold. Their antioxidant defenses are overwhelmed. The resulting oxidative crisis triggers apoptosis from within. In healthy cells with large antioxidant reserve capacity, the same curcumin exposure does not push ROS levels to a critical threshold. Instead, the antioxidant activity of curcumin dominates, reducing baseline oxidative stress and actually protecting those cells. This selective toxicity toward cancer cells while protecting healthy ones is the defining characteristic that has made curcumin one of the most studied natural compounds in oncology. Mechanism 5. WNT, signaling pathway suppression and cancer stem cell targeting. One of the most significant recent advances in cancer biology is the recognition that conventional therapies frequently fail to prevent recurrence because they successfully reduce the bulk of a tumor while leaving intact a small subpopulation of cells called cancer stem cells. Cancer stem cells are the cells capable of regenerating the entire tumor from a small starting population. They are the seeds from which recurrence grows. And cancer stem cells are uniquely resistant to the therapies that kill bulk cancer cells because they express higher levels of multi-drug resistance proteins and maintain active WNT signaling that confers self-renewal capacity. Curcumin has been shown in emerging research to target cancer stem cells through WNT signaling pathway inhibition. The WNT pathway controls stem cell self-renewal and differentiation throughout the body. In healthy tissue, it is tightly regulated to allow stem cell maintenance without uncontrolled proliferation. In cancer stem cells, WNT signaling is constitutively active, providing the self-renewal capacity that makes these cells so therapeutically challenging. Curcumin inhibits beta-catenin nuclear translocation, the critical step through which WNT signaling activates its target genes, effectively removing the molecular signal that maintains cancer stem cell identity and self-renewal capacity. While the human clinical evidence for this specific mechanism is still developing, the laboratory evidence across multiple cancer cell lines is consistent and compelling. Research published by the University of Michigan Comprehensive Cancer Center demonstrated that curcumin reduced cancer stem cell populations in colorectal cancer models by inhibiting WNT beta-catenin signaling, with the reduction in cancer stem cell frequency correlating directly with reduced tumor formation capacity in subsequent transplantation models. This mechanism matters for prevention because cancer stem cells are likely present long before a clinically detectable tumor develops. Dietary interventions that reduce cancer stem cell self-renewal capacity are therefore most relevant precisely in the prevention context, in the years before any diagnosis is possible, when the small populations of abnormal cells with stem-like properties are deciding whether to remain dormant or to establish the growth programs that will eventually produce a detectable lesion. Here is the protocol, and this is the section that most people get critically wrong. Curcumin in its raw form has approximately 2% oral bioavailability. That means if you consume 1,000 milligrams of curcumin, roughly 1 teaspoon of turmeric, your body absorbs approximately 20 milligrams. The rest passes through the gut largely intact. This is why so many people who have tried turmeric for health purposes and noticed no effect reached that conclusion. They were consuming the compound in a form the body could not meaningfully absorb. The fix is 2-ingredient and takes 3 seconds: black pepper and fat. Piperine, the active alkaloid in black pepper, inhibits the intestinal enzyme responsible for curcumin metabolism and dramatically slows its elimination from the gut, increasing curcumin bioavailability by approximately 2,000%, not 20%. 2,000. 1/4 teaspoon of black pepper added to your turmeric preparation transforms a supplement with 2% absorption to one with bioavailability approaching 40% at standard dietary doses. Fat solubility is the second factor. Curcumin is fat-soluble, not water-soluble. Consuming it with a fat-containing meal or dissolving it in a small amount of oil before ingestion further increases plasma curcumin levels by approximately 7 to 8 times compared to dry powder consumption. The protocol: 1 teaspoon of high-quality ground turmeric, approximately 200 milligrams of curcumin, combined with 1/4 teaspoon of freshly ground black pepper, dissolved in or taken alongside 1 tablespoon of coconut oil, olive oil, or the fat component of a whole food meal. This can be incorporated into cooking, stirred into hot beverages, or consumed as a direct preparation. Twice daily administration, morning with the first meal and evening with dinner, maintains more consistent plasma curcumin levels than a single large dose. From clinical practice, here are the critical details. Choose turmeric products that specify curcuminoid content. The compound you want is curcumin. Turmeric root contains only 3 to 5% curcumin by weight. Products standardized to 95% curcuminoids at doses of 500 milligrams twice daily provide substantially more reliable therapeutic concentrations than generic turmeric powder alone. If using powder, supplement with a commercial curcumin extract that specifies curcuminoid percentage. Avoid proprietary absorption-enhanced formulations that rely solely on synthetic emulsifiers. While some commercially available enhanced curcumin products show genuine bioavailability improvements, the simple black pepper and fat protocol achieves comparable results at a fraction of the cost and without synthetic additives. Do not heat turmeric above 170 degrees Celsius for extended periods. Brief cooking at lower temperatures and incorporation into dishes near the end of cooking is preferable to long high-temperature exposure, which degrades curcumin content significantly. Common mistakes. The first is consuming turmeric in capsule form without any fat or piperine. This is by far the most prevalent error and explains the persistent skepticism about curcumin among people who have tried it without effect. The second is inconsistency. The mechanisms described here, BCL2, downregulation, NF-kappa, B inhibition, PI3, K pathway suppression. Operate on weeks to months timelines at the cellular level. A 3-day trial tells you nothing. A 90-day consistent protocol allows the cellular environment to genuinely shift. The third is treating this as a substitute for appropriate cancer screening and medical care. Curcumin is a dietary modulator of cancer biology. It belongs alongside conventional prevention strategies, not instead of them. If you have risk factors for cancer or abnormal findings on any screening test, the appropriate response is immediate engagement with your medical team. Let us bring it together. Cancer cells survive because they hijack the biological systems meant to destroy them, specifically the apoptosis pathways that healthy cells use to self-destruct when they carry dangerous mutations. Curcumin, the active compound in turmeric, engages 5 distinct mechanisms that collectively restore the cellular environment in which abnormal cells cannot sustain themselves. BCL2, downregulation restoring the apoptotic program, NF-kappa, B inhibition eliminating the inflammatory survival advantage, PI3, K-AKT-MT, OR pathway, suppression reducing uncontrolled proliferation, pro-oxidant activity in cancer cells, pushing them past their oxidative stress tolerance threshold, and WNT beta-catenin pathway inhibition targeting the cancer stem cell populations that conventional therapies frequently leave intact, the cells most responsible for disease recurrence years after initial treatment. The protocol is 1 teaspoon of turmeric with 1/4 teaspoon of black pepper and a tablespoon of fat, twice daily, consistently. The bioavailability difference between consuming this correctly and incorrectly is not marginal. It is the difference between 20 milligrams absorbed and 400 milligrams absorbed from the same dose. One preparation produces a measurable biological effect. The other produces an expensive way to color your dishes. That difference is not a matter of preference. It is a matter of basic pharmacokinetics that determines whether the mechanisms described in this video operate at all in your body. Your cellular surveillance systems are working right now. They are identifying abnormal cells. They are initiating destruction sequences. The question is only whether you are giving them the molecular tools they need to complete the work. That bright yellow spice in your kitchen cabinet, the one that has been there the entire time, is one of the most documented natural activators of those systems in the scientific literature. Starting today, you have the information. You have the protocol. The decision to act on it takes less than a minute. Dr. Liana out. One additional consideration that deserves specific mention for this audience. Curcumin has documented interactions with certain chemotherapy drugs and blood thinners. If you are currently undergoing any cancer treatment or taking anticoagulant medications, discuss curcumin supplementation with your oncologist or primary care physician before beginning. In some contexts, curcumin may enhance the efficacy of conventional treatments. In others, it may require dosage monitoring of concurrent medications. Individual guidance matters here.